Well, hello, and welcome to this lab overview or introduction. As we come back from spring break, I want to make sure everybody's on the same page. When we last left, what we had accomplished inside lab is we had introduced the brain and the spinal cord. And remember, the way that I introduced them was pretty much as simple as possible. When you're thinking about, say, the brain, it's a ball, and spinal column is a stick. And then what we do, of course, is we layer on first some more general information, like there are 12 pair of cranial nerves here, and there are 31 pair of nerves that allow for sensory input and motor output, and each of these will have specific names. Okay, and then we actually have physical characteristics for both the brain and the spinal column, and that's what we focused on in the last lab. Okay, so what I want us to do now is think about, because your practical will be on Wednesday, I want you to think about how we layer now details and functionality onto this basic information. Okay? Let's do that. Let's go here. So prep for the lab. We're going to review what we just did before, okay? And I'm going to look at the lab packet. Hopefully it's completed, meaning you have finished off everything inside that lab packet that you can actually finish off. So when I see you on Monday afternoon, you're going to be able to sort of be, excuse me, familiar with a lot of the information we're going to be talking about. The goal of the lab itself is going to be to dissect primarily a brain. I will not give you the eye to dissect this time. We'll wait till later in the semester to do that. And then we're going to focus on learning the nuclei of the cranial nerves, which means what you're doing is you're learning where inside the brain the cell bodies of those cranial nerves are going to be found. Notice I said inside the brain. There's going to be a couple of sets of nuclei that are not found inside the brain, and that's important because that's what you're actually trying to learn here. You're going to learn the name, the number, and the function of the 12 pair of cranial nerves. And when we think about what's going to be on the lab practical, it's going to be sort of like what I had on the beginning slide there. It's going to be the brain, the spinal cord, and what are the cranial nerves. Remembering that we're about to introduce functionality. So let's get rid of these things here. Let's go to this next one here. So I told you ball and stick. Okay, so now notice my cartoon of ball and stick. Hopefully this is, even though it's really simple, I mean, honestly, this represents the basic structures, right? So here's a brain. Notice you can't tell if it's anterior or posterior. It doesn't matter. It's ball on stick. Ball equals brain, stick equals spinal cord, right? We want to start layering pieces of information on top of this, okay? I'm going to focus first on the spinal column. Look at over here. Nature Reviews Neuroscience paid somebody lots of money to put a pretty little image together here. And if you can draw that, knock yourself out. However, notice there's still 31 pair of nerves if I drew them here. Doesn't matter. What's inside my cartoon here, of course, this is the end of the spinal cord. So that's the conus medullaris, right? CM. And any of the nerves that actually come off and head down are the cauda equina. So those are CE. And you can see what I'm doing, right? I'm trying to figure out really simple ways to remember all these basic structures. I'm going to draw an enlargement here, and I'm going to draw an enlargement down here. And when I do that, you know that that's our cervical enlargement, cerv, ENLG, LG, and likewise LUMB. And I'll leave out the B. ENLG. Right? Okay. Fantastic. Now, let me switch some colors here for you. Go to blue. Let's not go blue. Cranial nerves. Let's give me some fancy colors for cranial nerves. Fuchsia. Fuchsia. 12 pair. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 pair of cranial nerves, right? Yay. Now, inside your mind's eye, remembering that's happening on both sides. That's why we say 12 pair. if we're thinking next, so we got 12 pair of cranial nerves, we've got 31 pair of somatic sensory and motor nerves. We have our cervical enlargement, we have our lumbar enlargement, we have our conus medullaris, we have our cauda equina, the pia mater from the conus medullaris traveling all the way down to the bone, of course, makes the filum terminale. TRM, right? Term. Yes, no, maybe. Keep it this simple when you're reviewing it. Actually, for that matter, keep it for this simple when you're first learning it. And then we're going to, of course, as usual, add more and more layers of detail. And where's the layer of detail here? Well, I could switch colors one more time. Let's go to this sort of, I don't know if you can see this. Okay, I apologize if you can't. Okay, you're going to have a cervical plexus. Yes, no, maybe. Okay, and then maybe out here you'll have a brachial plexus. And then down here on both sides you'll have a lumbar plexus. And then down here you're going to have sacral plexus. And you'll have a coccygeal plexus. Okay? And you'll notice it's not inside the thoracic region. And that's the sort of crap you need to pay attention to. Okay? So hopefully this looks fairly familiar, and hopefully when I see you studying for your exam, you can draw something like this. I'm not even just talking about your practical. When you're thinking about how to apply these really simple pieces of information, if you drew this, this can help you answer questions on your exam. You'll see what I mean in a few minutes. Okay, so what else can we actually add to this cartoon? Well, you'll notice that we haven't talked about what's anterior, what's posterior, anything like that. But going down the front of the anterior surface of this, which would be over here, right? That would be the anterior surface. You can see here that would be the anterior median fissure. And that's what happens when you cut the stick. Now you can start looking at who's anterior or ventral, right? So I'll put a line over here, go ventral, and I'll go a little line back here and go dorsal, right? And here's your dorsal root ganglia. That's how you know where you are always. D, R, G. Okay, cool, fantastic. You notice these are colored. Okay, color means something. Anytime an artist takes time to actually fill in some color for you, they're trying to communicate something that's universally shared inside all of these disciplines. And blue, for the most part, is always going to be thought of as, in this case, these look like they're all motor outputs, but they're lying to you, because inside our textbook, it's supposed to be sensory input, and in red is supposed to be motor output. So even though they're supposed to communicate something, I don't think they're really being specific here, and you'll see what I mean when I talk about it inside class. The one thing I do want you to see, though, is look, take a look at this white matter. That's all myelinated axons of neurons, right? And that means gray matter here are the cell bodies of neurons as well as unmyelinated interneurons. And that's what makes up the gray matter of your spinal column. That also means it's going to make up the gray matter of your brain, but we see why it's different in a few minutes. Okay? Cool. Let's get rid of some of these colors. Let's move on to the next slide. So how are we going to add on to this information? Well, Mother Nature takes our little ball and stick, okay, ball, stick, green. We have our ball and our stick, and what she's going to do is she's going to, let's say this, we're going to say Mother Nature has figured out a way to take sensory inputs, convert it into, you need a purple here, something that needs to be considered for just a moment or two, and then you have to act on it, right? You think about if I ever picked up something and threw it at you, or somebody came up to you and threatened to hit you, what would you do? You'd act reflexively. And that means when we put in our pieces of information here, neutral color here, we're going to have some sort of sensory input, that's SI, and it's going to be interpreted here in our interneuron, okay? Inter. And that interneuron then determines which motor neuron activates what musculature. Okay? So we're going to say this is a motor neuron. Motor neuron. Okay? And that means that there are going to be, so they'll have some sort of an effector here. Effect. And so that means, in reality, you have some sensation. It's going to be collected at a sensory receptor, SR. Sensory receptor is number 1. Sensory neuron transmits, okay, or afferent neuron, number 2, transmits that information. I'm going to put A for afferent neuron here, to our interneuron. And our interneuron is inside the gray matter, inside our spinal column. Okay? And that's number 3. Remember I said our interneuron considers for a moment what's going to happen. Remember, this is not myelinated. So let's make sure you guys understand that. Remember, these circles represent myelination. That means myelinated neuron signals to non-myelinated neuron. Non-myelinated neuron signals to myelinated neuron. And that's going to actually exit out of the spinal column. So let's add one more layer of information here. If this is traveling into the spinal column, it means it's traveling into the dorsal root ganglia, and the cell bodies of those sensory afferent neurons are located inside the dorsal root ganglia. And that means it's always going towards the back of the spinal column. This interneuron is then signaling for motor output to exit, and that means it's always going to be exiting the ventral portion or the anterior portion of the spinal cord, either on the right or the left side. It makes no difference. But the significance here is, look, we took a really simple cartoon that has 3 colors to it, blue, purple, and red, and those blue, purple, and red represent what we see inside the textbook, so that you can understand this thing that's called a reflex arc. And it has now, to finish this off, if we've activated our motor neuron, we have 4 parts. The important part is that there are 5 parts to your average reflex arc because you have to activate an effector. So let's just go over this again. Sensory receptor on our afferent neuron signals to our interneuron. Our interneuron figures out what to do, and then we activate our motor neuron, MN, to exit through the ventral portion of the spinal column to activate or target our effector. Okay? Yay. Cool. What we're going to do in lab is we're going to look at somatic tests. We're going to hit each other inside the knees. Autonomic tests. We're going to flash light bulbs into each other's eyes. Okay, I don't think we have time to do some of the other tests that can be done inside this class just because of time. Okay, fantastic. Let's switch gears here. I hate saying that. Oh no, what is this? I want you out of here. Hey, pause. Sorry about that. It looks like, what is it? Adobe's being imperious, trying to interrupt our presentation. Let's finish this up. So here's your brain, right? Okay, here's your brain on class. And what I mean is you're thinking about the fact that we're going to be dissecting a sheep's brain, but here's what a human brain looks like. One of the places I want to start us off with is you're going to notice it has this sort of covering on it. That's pia mater. Okay, and if you think about what the meninges were, okay, remember you have the dura mater. Get some coloring here. I don't need coloring. Dura mater is attached, so go D, is attached because they're actually webbed together to arachnoid matter, which is why I'm drawing that little line there, so that gives us an A. Then we have a subarachnoid space where I probably should change the color to cerebral spinal fluid, which I'm going to make blue. Let me make it a light blue. There you go. Just for educational sake. Yay, light blue represents cerebral spinal fluid. And let's go back to the black, where now we have pia mater. Okay, and that pia mater is attached to our nervous system structures. And that's why it has a sort of like whitish, sort of opaque color to it. So what are we going to do with this information? Well, chances are you're going to get a sheep's brain that has all of these attached to it, and you're going to have to cut through it. So let's take that sheep's brain. We're going to look at it from the superior view, bird's eye view. It looks something like this. All right, and we're going to cut. Sorry about my bad drawing. We're going to cut right down the middle on one piece of it, right? And then you're going to take one piece, meaning the resulting 2 halves. You'll take 1/2. It doesn't matter which half it is. And I want you to then to do a frontal cut on it. So do mid-sagittal, and then you do a frontal cut. In doing so, you're going to reveal both the basic obvious structures we saw on the plastic models inside lab, as well as the internal structures you were not able to see inside lab. Okay? Now, the last thing I want to introduce is, okay, now, yay, we need to learn these cranial nerves. And everybody has their favorite little mnemonic, you know. Some of them are a little more racy than others. Whatever. I don't care how you learn it as long as you learn it. But what I probably focus on more than anybody else does is where do we find the nuclei of these cranial nerves? Because where the nuclei are where the nerves are actually executing their responsibilities. And that's what's really important for the rest of your lives. It's not, can you memorize 12 paracranial nerves? That's important to do, and I expect that you do it. But let's take a quick look here. Notice that the olfactory tract is actually located outside of the brain. So the nuclei of these cranial nerves will be found where this is working. Likewise, our optic nerve, it's actually found outside of the brain. So therefore, its nuclei of these cranial nerves is found outside there as well. However, once we start getting down here and we get to the midbrain, we can see the trochlear, the trigeminal nerve, okay, as well as the abducens. No, not abducens. You can see that in the midbrain here, we actually have, there it is, ocular motor. Okay, these are all functioning to regulate the midbrain responsibilities, and you're going to find the largest number of nuclei there. Skipping over the pons, let's go take a look at the medulla oblongata, okay? So you have the hypoglossal, you have the vagus, you have the glossopharyngeal. It gets a little bit tricky when you're looking at the vestibular cochlear as well because it's actually inside both. And then you can see as we move on to the facial and the abducens, we've split the responsibilities. And it's really key that what I told you inside class and lecture, actually lab, to learn where the cell bodies are located so that when we get into chapter 15 and chapter 16, you can understand why they are where they are. Okay, so I've been talking for too long. Hopefully this gives you an overview of what we're going to be doing in lab the next time I see you. Have a great night. One more thing. I just signed off and thought about it. Your practical will be on Wednesday, so this is why it's important you have to finish that packet, right? That being said, I know we only have a limited amount of time to put all this information inside there. So if you pay attention to what I'm talking about here, you will have a pretty good idea of what I'm hoping you will be looking at on the practical on Wednesday evening. Okay, have a good night now. Bye.